Welcome back to my class again. So we were discussing NCBI and its different resources. So far we had discussion on database retrieval tools. We discussed BLAST, and today we'll be moving on to the resources for gene level sequences. So under these resources for gene level sequences, the first one we are going to discuss is Unigene, then Homologene, and third one is RefCQ. As you all know, ESTs are expressed sequence tags. These are actually certain markers or tags used in the process of sequencing, especially in the sequencing of genome. Unigene is a database that actually provides information about different ESTs that belong to a gene. On a particular gene, there will be different researches going on, and at different laboratories, they will create different ESTs. They may belong to one gene. So this Unigene help us to identify those ESTs which are referred to a particular gene. So you can actually make out the answer from this word uni. Uni means one. So all the ESTs belong to one gene can be easily identified using database Unigene. Homologene is a database of orthologs and homologs. See, the orthologs and homologs of human, mouse, rat, zebrafish and cow gene, they are actually present in the homologene. They are having information about orthologs belonging to human, mouse, rat, zebrafish and cow. RefCQ is a curated database of mRNAs and proteins. These mRNAs and proteins of organisms like human, mouse and rat. RefCQ is a database which is having information of mRNA and protein of different organisms. Homologene tells us about orthologs and homologs, and Unigenes tells us about ESTs belonging to a particular gene. Some of the databases which are given in your textbook are EMBL, European Molecular Biology Laboratory Database, and this database also containing a large collection of nucleotide sequences. This is also a resource level database which provides information on various which provides information of genes, mRNA, proteins, etc. This is also like NCBI, another big information database. Okay, European Molecular Biology Laboratory. UniProt is a protein database. As the name suggests, it gives information about proteins. They are also known as SwissProt. These informations are annotated protein sequences. Annotated means their entire characterizations are done. All the information regarding a protein is being studied, and they are curated and they are stored in this database. So UniProt, you can see in UniProt, especially they are meant for proteins. Another resource level database. See, PDB is Protein Data Bank. They provides us three-dimensional structure of proteins. You can have three-dimensional structures of proteins You can even analyze, search, visualize the three-dimensional structures of proteins. See 3D structure of proteins. You can have different protein sequences and their 3D structures. Okay, so if you want to check any structure of any protein, you can go and check their structure using protein database. For example, if we go chymotrypsin A and B chain. See these many chymotrypsin and their varieties are present here. You can have the three-dimensional view. PDB is an information level database. This database provides us information exclusively about ribosomal RNAs, rRNAs or ribosomal RNAs. Okay, it says special database which is dealing with ribosomal RNA. Phylogenetic analysis, phylogeny, and alignment of homologous proteins. This is PALI database. This database gives us information about the proteins and their phylogenetic. Phylogenetic means their evolutionary informations. Now in the last part of your chapter is dealing with what are the analysis that we can do by using bioinformatic tools. सबसे पहले we can have process raw information, processing of raw information. Raw information means, suppose if you get after experiment some sequences, you just want to know whether it is RNA, is it DNA, or is it rRNA, or is it is actually mRNA, or whether it is tRNA. All these information can be done by using certain bioinformatic tools. They also help us to identify whether it is regulatory part of the gene, are they a part of intron, are they a part of exon, etc. Second is we can have gene prediction. That means if you have a set of sequence with us, we can predict whether there is a gene present or not. Okay, two names are given in your textbook, that is gene mark. Gene mark is for bacterial genomes and gen scan. This is used in eukaryotes. See, this is gen scan. Here you can paste your sequence and find whether there is any gene or present or not. Okay, if you know the organism, then you can identify that organism, or you can give that organism, and you can predict the present number of genes and all. This is gen scan. Gene mark is for bacteria and gen scan for eukaryotes. Now this has been actually modified. You can also go gene mark ES, which will tell you about gene prediction in eukaryotes also. That has been modified. Okay, many modifications are happening on these databases day by day. Okay, we can have gene prediction. Then we can have information about proteins. If you have gene sequences, you can find mRNA, you can find the corresponding proteins or amino acid sequence. You can predict by using softwares. Then also you can have information of or regulatory sequences. If a gene sequence is there with you, enhancers and silencers can also be identified using bioinformatic databases. Phylogenetic matlab evolutionary. Phylogenetic informations can be, or relationships can be identified using various databases. Like PALI, we had a discussion on PALI. This is a database exclusively for that. You can also have a discovery of certain unknown genes by using various databases present in bioinformatics. So altogether we can say the first one is processing raw information, second one was prediction of genes. You can have information regarding proteins, we can have information regarding regulatory sequences, we can have information on their phylogeny, and you can also have certain discovery using bioinformatics tools. So altogether bioinformatics helps us in different ways, in various ways, for present research. Thank you.